For me, it's
How about the others?
clear, Doctor.
Okay.
Boleh, Doctor.
Alright.
Juga, Doctor.
Okay. Saya kena menjerit sikit lah dalam office ni kan. Okay. 44, not bad. Alright. Let's continue on our lecture last week. So before we start, so how was your test 2? Okay, test 2.
Rough, Doctor. Very bad. I don't know what.
Kalau Dr. Dina selesai.
Alah, Doktor, dua-dua kot. Saya pun tak tahu apa yang saya jawab.
Inilah, saya pun belum dapat exam paper lagi, mungkin by Tuesday kot Dr. Dina kampas kan. Okay, so last week, siapa ingat kita stop dekat mana? Hey, this one RNA, kita boleh sama-sama. So not this one. This one. Yeah. Okay, boleh nampak slide saya?
Nampak? Kalau Doktor.
Okay. Saya tak buka kamera lah hari ni sebab nak kena dekat dengan ni. Nanti penuh pula saya punya kamera kan. Alright, so kita start dekat mana? Apalah. So I think I will continue with this slide. Okay, kalau kita dah discuss pun, but it's okay. You can just continue. Boleh revise balik. So I would like to start with this slide. So on the mechanism of DNA replication. Okay, but I would like to remind, kalau tak clear, bergema ke apa ke, just inform me. Alright, I'll try my best to improve. Okay. So,
Doctor.
okay, thank you. So, alright, for mechanism of DNA replication. So these are the general features of DNA replication that you need to know. These are the terms. Okay. But by having all these terms, apart from knowing all the terms related to DNA replication, you need to understand what does it means by all these terms. Okay. First of all, okay, we know that DNA replication is a semi-conservative. It shows semi-conservative characteristics. So what does it mean by semi-conservative? Anyone? Biasa dengar kan semi-conservative. So what does it mean by semi-conservative? Who can explain? What does it mean by semi-conservative? Okay, semi-conservative is okay, we know that DNA it's a double-stranded DNA. Okay, so it separates into two single strands, okay, that act as a template for the formation of a complementary strand. Okay, so semi-conservative tells you that one strands are formed from the original template of double-stranded DNA, and another strand is from that is a newly synthesized strand. So when you see the term of semi-conservative, it tells you already understand that that the double-stranded DNA, that is a newly synthesized double-stranded DNA. One strand is originally from the template, and another strand is a newly synthesized strand. So it gives it explains the term semi-conservative. So how about semi-discontinuous? How about semi-discontinuous? Anyone?
Replicate by Okazaki fragments.
Okay.
Discontinuously.
Semi? Okay, semi-discontinuous. Okay, that one is true, correct. But when we talk about semi-discontinuous, by having the word semi, it tells you that leading strand is synthesized continuously, but for lagging strand is synthesized discontinuously by having Okazaki fragment. So that's why it's called semi-discontinuous. Okay, because for leading strand it is synthesized continuously. Okay, and for DNA replication, it involves complementary base pairing. Okay, once double-strand DNA is unwind into two single-strand DNA, so that single-strand DNA acts as a template. So DNA polymerase as an enzyme that add nucleotide that complement, okay, to that nucleotide at the original template to form new strands. So complementary base pairing, I'm sure you know, A is complement to T, G complement to C. Okay, guanine to cytosine, adenine to thymine. Okay, DNA replication is asymmetrical. So what does it mean by asymmetrical? Anyone?
Not symmetrical.
Okay, so what does it mean by not symmetrical in terms of DNA sequence, in terms of the mechanism of DNA replication? Not asymmetrical, it tells you that, okay, kalau in DNA structure itself, DNA run in opposite direction. So it tells you that it's asymmetrical. So in DNA replication, it inherit, okay, the DNA sequence structure as asymmetrical. So it inherit, okay, the mechanism inherit the asymmetrical in terms of DNA mechanism. Okay, and replication occurs in 5' to 3' direction. So anyone can explain this? So how does this replication occurs at this specific location? 5' to 3' direction. Kita dah discuss dalam nucleotide metabolism. In between apa? Okay, 5' is if you remember the structure of nucleotide, 5' is where the phosphate, okay, phosphate located, and it react with 3' of another nucleotide. It react with hydroxyl group of another nucleotide to form phosphodiester bond. Okay, that make a backbone for DNA structure. Okay, so I think I have explained this. Okay, the process, the mechanism of DNA replication. So it's a tightly controlled process. It occurs at specific times during the cell cycle. It requires a set of protein and enzyme. It requires energy in the form of ATP. So there are 3 basic steps: initiation, elongation, and termination. And one of the most important basic components that needs to have in order to perform replication is template and primer. So basically, all this theory, okay, it apply when we perform polymerase chain reaction, which is PCR. If you want to make a copy of E. coli gene, if you want to make a copy of any plant gene, okay, so it requires this, okay, it requires a protein and enzyme, it requires, so it will undergo all these 3 steps. Also it needs a template So that DNA sequence, it needs a primer, it needs enzymes, okay, to aid, okay, the replication process, okay, through PCR, okay. If you have the opportunity to conduct PCR dalam lab or during your FIP, so it all this theory will apply. So you need to understand what are the enzymes needed. So what are the proteins? What does it mean by initiation, elongation, termination? Okay, initiation meaning that from double-stranded DNA, so we need enzyme to break, okay, to break the double-stranded DNA into a single-stranded. So elongation, so is a process where DNA polymerase comes in and add a nucleotide that complement, okay, to the DNA in the original template. So it synthesizes new strands, okay. Then later, after specific cycles, for example, 30 cycles, 28 cycles, or more than that, okay, cycles. So we need to stop, okay. So it will undergo termination process, okay. So these are the most important basic components. We need to have template. Template is the DNA sequence, okay, and also primer. Okay, primer is something needed to start the DNA replication. Okay, kalau dalam PCR process, we need to design a primer, meaning that once we have the DNA sequence, okay, that specific contoh, okay. We can download, okay, sequence of the genome sequence of E. coli, for example. But we want to focus on cell wall binding protein. Kita nak amplify cell wall binding protein of E. coli, okay, for example. So we need to find that specific sequence for cell wall binding protein dalam the whole genome sequence. Contoh, hanya daripada berapa kilodalton punya sequence of DNA, mungkin we want to amplify 300 base pair of cell wall binding protein. Okay, so that 300 nucleotide sequence, okay, kita cari the so from that we need to design a primer to help in the amplification process of that specific gene. So we need this primer. Primer kalau dalam PCR we need to design by ourselves, but it depends on what kind of application that you are going to carry out. Okay, ada certain kalau you buat 16S, 18S, biasa dengar tak 16S RNA, 18S RNA, biasa dengar tak? Do you know what does it mean by 16S, 18S? Tak? Okay, it's about genome. Okay, kalau 16S, it more applied to bacteria. Okay, maybe you need to look to do a reading on about bacteria ke E. coli ke about the genome of this microorganism. But kalau 18S, it applies to eukaryote system. Okay, eukaryote system is more complicated, so dia tak sama. Okay, genome structure of eukaryotic system is not similar dengan prokaryotic. Prokaryotic system is much simpler, sebab tu dia hanya ada 16S. Okay, 16S explain on the structure of the genome. Okay, kalau you study on 18S, dia lagi complicated. Alright. Okay, so okay, apart from contoh lah kalau dalam PCR kan, apart from this, okay, one of the most important one, okay, kita kena ada DNA polymerase lah. DNA polymerase, this is part of the enzymes. Okay, DNA polymerase also known sebagai DNA replicase. Okay, okay, selain DNA polymerase itself, it tells you that it helps to synthesize, it helps to add nucleotide for the complementary base pairing and synthesize new strands. Okay, so these are the enzymes that extend the primer. Okay, once kita ada primer, kita design primer, so it helps to extend the primer for the synthesis of new strands. Okay, so basically sebenarnya DNA polymerase ni there are few types. Okay, there are polymerase 1, polymerase 3, and also polymerase 2. Okay, but polymerase 1 is more on filling the gap, okay, between the newly synthesized Okazaki fragments. We know that in lagging strand, okay, in lagging strand, you cannot synthesize continuously as leading strand. So it's synthesized by having short fragments, which is Okazaki fragments, and there are gaps between every segment, there are gaps. So polymerase 1 helps in filling the gap between the newly synthesized Okazaki fragments. Okay, but for polymerase 3, so polymerase 3 is the one that helps in the replication process. It helps in producing new strands of complementary DNA. Okay, so polymerase 3 is the one known as a replicase. Yep, replicase juga lah dia dikenal sebagai. And one thing that you need to know is about if you have a look, kalau ada kesempatan untuk baca journals on DNA polymerase, okay, even though we have a variety of DNA polymerase, as I mentioned just now, we have DNA polymerase 1, 2, and 3, but three structures suggest that they share a common catalytic mechanism. Okay, all these variety DNA polymerase, they share a common catalytic mechanism for nucleotidal transfer, which their active sites all contain 2 metal ions, which is normally is magnesium ion. Okay, they share all variety of DNA polymerase. They share, they have their active sites have 2 metal ions, which is magnesium ions. Okay, walaupun kita ada variant, we have variety of DNA polymerase, but they have common catalytic mechanism, which is the active sites contain metal ions, which is magnesium ion. So what does this metal ion, what is the role of this metal ion? Okay, so this metal ion, it helps to activate the primer. Okay, it helps to activate the primers, especially at the 3' hydroxyl group. Okay, for the nucleophilic attack on the phosphate group. Okay, kind of replication, it happens at the 5' and also 3' end. So that's how DNA binding to each other. That's how nucleotide bind to each other. So these metal ions help to activate, okay, the primers at the 3' hydroxyl group for a nucleophilic attack on the phosphate group, 5' phosphate group. Okay, all right. So apart from DNA polymerase, so there are additional enzymes or proteins that required in DNA replication. Okay, we need DNA helicase. So this enzyme that helps to break down the double-stranded DNA. Okay, and we do have single-stranded binding proteins that keeps the DNA helix open. Okay, bila dia dah jadi single-stranded tu, if there is no the single-stranded binding protein, it tends to anneal, to re-anneal. Okay, so we don't want that to happen because we need single-stranded to keep the helix DNA to keep open for the replication for the DNA for the replication process to occur. And also we need primase, okay, primase. So in our system, we do have primase because it helps to create RNA primer. Sebab kalau kita buat PCR, kalau kita yang buat PCR, we can design our own primer. But in our system, dah cantik dah. Okay, we do have primase that helps to create a primer. Okay, for the synthesis of DNA. And last one, we need to have ligase. Okay, ligase is an enzyme that join two fragments together, that helps to bind, that helps to weld. Okay, two Okazaki fragments. Okay. Okay, so this is the general figure to explain about DNA replication. Okay, from double-stranded DNA, we do have helicase that helps to unwind the double helix. Okay, once helicase break down this double-stranded DNA, so single-stranded binding protein comes in to help, okay, to keep the helix open. Okay, it helps, it prevent the degradation of the DNA. Okay, itu satu. Okay, and also it makes sure that DNA helix keeps it prevents from the re-annealing of the DNA helix. And also we have this primase. Okay, bila dah menjadi single-stranded, so it acts as a template. So primase comes in, primase help to synthesize RNA primer. So kita ada DNA polymerase 3. Okay, so this is where DNA polymerase 1 comes in. It helps to fill in the gap between the Okazaki fragment dekat lagging strand. Okay, DNA polymerase 1 dia mesti ada dekat lagging strand, not leading strand. Okay, and same goes to DNA ligase. Dia nak weld together all the Okazaki fragments. Okay, so how about topoisomerase? You all dengar saya tak? Clear tak?
Dengar doktor, tengah saya
Dengar.
Right, so how about topoisomerase? Anyone?
sekejap doktor, tengah saya.
Is it prevent tightening DNA strand bila dia buka? Lupa lah.
Okay, yeah, it could be. Okay, that's why nama dia topoisomerase. Okay, it helps to unravel, it helps to overcome, to solve any topological problems. Contoh ada twist of DNA ke, kan bila from double-stranded DNA, so helicase breakdown the double-stranded jadi single-stranded, but they may face a problem, ada twist kat DNA structure ke, here and there ke. So topoisomerase dia akan ada di situ to help to solve all the topological problem. Okay, whatever problem occurs during sebelum dia binding ke apa, they help to solve the problem, so that helicase boleh break down the double-stranded DNA, dia boleh unwindkan the double helix. Okay, so that single-stranded binding protein dia keeps the double helix open. Okay, the single-stranded open. Okay, alright. So this is something, the figure that helps you to understand what does it mean dengan kat atas ni lah. Okay. Alright. So Okazaki fragments. Okay, so what is Okazaki fragments? Okay. We know that leading strands is synthesized continuously. Okay, because DNA replication occurs from 5' to 3'. So it cannot synthesize from 3' to 5'. Betul? Okay. From 5' to 3'. But kalau kita tengok kat sini, lagging strand is since it cannot synthesize because dia dekat opposite direction. So it cannot synthesize continuously seperti leading strand. So how this lagging strand is synthesized by having short lengths of DNA as an opposite. So it is synthesized by as a short length of DNA. Okay, as a Okazaki fragments, okay, that are produced by the discontinuous replication of the lagging strand. Okay, so how does this Okazaki fragment synthesize? So by primase kan. So primase ni dia akan producekan RNA primer. So once we have this RNA primer, dia akan synthesize this short fragment lah. Okay. So these are the range of length of this fragment. Okay, kalau dalam bacterial cells, it's up to 2000 nucleotides. In eukaryote is much shorter compared to bacterial cells. Okay, is kadang-kadang dia hanya up to 200 nucleotides. Okay, because eukaryotic cells, the system is very complicated. Okay, even though scientists has worked in human genome project for many years, but it's still not fully understood. Okay, there are many other things that need to explore on human genome project. Even though they can sequence a human genome, but there are many other parts that need to be studied, that need to be explored in order to understand the whole system. Okay, banyak lagi. Okay, it's very complicated one. Okay, so once these Okazaki fragments synthesize so that the DNA polymerase helps to filling the gap in between the Okazaki fragments, and ligase helps to combine, to weld together all these Okazaki fragments to form this lagging strand. Okay, so it's about this one is about DNA helicase. Okay, so this enzymes that unwinds DNA at a position called origins. Okay, so this is where the replication occurs. Okay, where the synthesis will be initiated. Okay, so this enzyme moves 1000 base pair per second, kind. Okay. And once the DNA helicase break down double-stranded DNA, so we have the single-stranded binding protein, helps to protect the single-stranded DNA from degradation and secondary structure formation. So it also prevents the re-annealing of the single-stranded DNA menjadi balik double-stranded. Okay. Regions of replication. Okay, so this is where this is location. So this is where kita punya cells dia tahu kat mana nak mulanya replication. So it identify the origin. Where is the origin of the replication? So our cells can identify. Okay, contoh kalau macam saya nak buat di specific sequence ke dalam E. coli punya system. I need to amplify that specific sequence. So in order untuk saya clone ke dalam E. coli system, saya kena tahu kat mana origin that E. coli system. I need to know origin ni dekat mana, dia punya cloning site di dekat mana. So I need to know that. Okay, because kalau dalam punya system, our cells can identify. Okay, we have specific location where the replication process can be initiated. Okay, so these origins can be recognized by the sequence. Okay, so kalau bacteria, it has single origin. Okay. On its chromosome. Kalau eukaryotic system is more complicated, dia akan banyak, dia akan ada multiple origin. Okay, so for prokaryotic system bacteria, the origin is about 245 base pairs long, and it it AT rich. Okay, what does it mean by AT rich? Okay, dalam 245 base pair, dalam nucleotide untuk 245 base pair ni, it contain mungkin more than 50% daripada 245 tu adalah adenine and thymine. So that's why it tells you that it is AT rich. Okay, tapi kalau kita nak design primer untuk amplify apa-apa sequence, kalau selain daripada you perlukan template, you need enzyme, you need, bila design primer, design primer, one of the important rule, you have to design a primer that contain more than 50% of GC. So it should be a GC rich primer. Kenapa? Okay, origin. The important point that you need to know, we are required to design a primer that contain 50% of GC dalam because we hope once because we want to synthesize a new primer, we want to make a copy of DNA, kan, kita nak synthesize DNA yang baru, we hope that it have a very strong strong DNA, because GC, we know that bonding between GC and AT, GC is much stronger because dia ada tiga hydrogen, dia ada 3 ikatan kan compared to AT. So we hope we want to synthesize a very solid, a very strong DNA without any mutation, without any, so it tak boleh sesenangnya nak di breakdown compared to AT lah. Tapi origin is a AT rich sequence, okay, in the 245 base pairs. Okay. Alright, so eukaryotic system, it has multiple origins, okay, of replication. Okay, kalau dalam bakteria, kalau apa-apa origin of replication, kita kalau dalam sistem bakteria dia akan ada ditulis ORIC, you automatically understand, so this is the origin of replication. Okay, origin of replication. Okay, so replication fork. So replication fork is what does it mean by replication fork? Okay, so replication fork is a situation that occurs after, okay, bila helicase dah breakdown the DNA, dia akan terbuka kan? Dia akan terbuka menjadi 2 kan? So from there, that Y-shaped structure tu, that situation is known as a replication fork. Dia macam fork sikit kan? Okay, so bila berlakunya di situ, dia sebenarnya it also making up a replication bubble. So dekat tengah-tengah tu dia akan berlaku replication bubble for the DNA replication synthesis. Okay, so replication forks will move in opposite direction as replication proceeds. Okay. Okay, a little bit on single-stranded body protein. Okay, prevent the single-stranded DNA from reannealing and protect it from nucleases. Okay, DNA must be stripped of this protein before it can be replicated by DNA polymerase. Okay. Okay. Okay, a little bit on primer and primase. Okay, primase enzyme kan, kalau ASE enzyme, so it's enzyme that makes RNA primer. Okay, or short stretch of nucleic acid complementary to the template. So once we have this primer, so the DNA polymerase can only add nucleotides to the 3' end of an existing DNA strand. Okay. Okay. Okay, about reading and lagging strands. Okay, important point you need to know, the synthesis occurs in 5' to 3' direction by the DNA polymerase 3. Leading strand is synthesized continuously while lagging strand in segments. Okay, and the segments are called Okazaki fragments. Okay, so DNA ligase helps to join together all these Okazaki fragments to form a longer DNA strand, which it helps to catalyze the formation of phosphodiester linkages. Okay, leading strand can be extended from one primer alone. Okay, that one is unique for leading strand, but for lagging strand, it needs a new primer for each of the short Okazaki fragments. Setiap Okazaki fragments too, it requires a different primer. Okay, so that's differentiate. Okay, that's this point differentiate between leading and lagging strands. Okay, so this is why explain the term of replication fork. Okay, replication fork. Okay, it's continuous. So which one is leading strand? Which one is lagging strand? Okay, ligase. Okay, how does this ligase helps in join together different fragments, Okazaki fragments to form a longer strand of DNA? Okay, so basically ligase helps to catalyze the formation of phosphodiester bond by an attack of 3' hydroxyl on the 5' phosphoryl group. So this is the mechanism that catalyzed by DNA ligase. So basically in DNA ligase, so this reaction requires the free energy that obtained from the coupled hydrolysis of NAD plus or ATP. Okay. All right. So this one, okay, kalau you tengok dekat sini kan, this is another figure that explain on the replication of E. coli DNA. Okay, helicase is a general name of the enzyme that unwinds the DNA, but it has their specific name, DNA-B. Okay, kalau you sebut DNA-B, dia dah tahu dah, this is helicase. Okay, helicase. Alright. So you can have a look at this figure on the role of each enzyme. Okay, so let's move on the DNA damage. Okay, kita dah discuss on DNA replication. Okay, what is Okazaki fragment, what is leading lead, what is Okazaki fragment, leading strand, lagging strand, the role of helicase, primase, what is primer, what is DNA polymerase, how ligase helps in joint Okazaki fragments. So now let's move to DNA damage. Okay. Okay. Okay, DNA, even though DNA replication is a very accurate process. Okay, a very tightly controlled process. So we need a very precise process to ensure that we don't we are going to synthesize a correct sequence of DNA. But DNA is still susceptible to damage from variety of sources. Okay, it's susceptible to damage from a variety of sources. Okay, one of it, kalau ada damage pada DNA, normally kita guna term mutagenicity. Okay. Kalau damage DNA, kita akan check pada dia punya mutagenicity. Okay, which is kalau mutagenicity is related to carcinogenicity that can be tested in living cells. Okay, so DNA replication, as we discussed, is dia ada proofreading mechanism. Okay, because it is essential for the accurate transmission of genetic information during cell division. Kita sangat-sangat, it is a very important okay, to have a very accurate DNA replication, but still DNA is susceptible to damage. Okay. But still DNA damage can be mended. Okay, can be mended, can be undergo alteration, but still it may be imperfect. Okay, kalau DNA damage, okay, because it can be bila dia imperfect, dia akan hasilkan mutation lah pada gene. Okay, so kalau if you look at this slides, okay, error rate for DNA damage, so it's a very rare, it's very rare berlakunya DNA damage ni. Okay, kalau contohnya in E. coli, 1 in every 10 power of 10 base pairs incorporated. Apa maksudnya? Okay, so kalau DNA replication, during the replication, DNA polymerase is aiding new nucleotide for the complementary base pairing. It is very rare that there is incorporation of new nucleotide. Even addition of one nucleotide to the sequence, it is very rare. Okay, because addition of one nucleotide either spontaneously oleh that affected by environmental factors, contoh UV, ionizing radiation, or chemical agents, or so all these environmental factors can physically damage DNA. It may cause addition of new nucleotide, it may cause of deletion of nucleotide, it may cause of the changing of the nucleotide. Contoh, okay, you are working in the lab. Contoh, kalau dalam lab sebenarnya banyak je chemical that harmful to living cells. Contoh, kalau kita nak ranjal, biasa you all pernah run agarose gel tak dalam lab? Mungkin second year belum lagi kot, kan? Ke dalam enzymology, saya rasa ada kot. Pernah tak ranjal?
Pernah.
Ranjal untuk tengok. Pernah? Okay. Kalau zaman dulu, zaman saya lah, zaman sekarang mungkin different dia punya chemical yang guna. Zaman saya dulu, untuk stain gel tu, in order to visualize, untuk kita boleh visualize gel tu bawah UV, we need to stain the gel dengan ethidium bromide. Pernah dengar ethidium bromide? Have you heard ethidium bromide before? Tak pernah? Tak, okay. Zaman sekarang mungkin berbeza lah, mungkin dia dah improve. Tapi zaman dulu, kalau nak tengok gel di bawah UV, kita kena stain gel tu dengan ethidium bromide. So basically ethidium bromide ni, it carcinogenic, it classified as carcinogenic. So you have to be careful. Kalau nak handle, nak buat benda tu memang pakai glove tu 2-3 lapis kan. So it can induce, so kalau kena, it may cause damage to DNA. Contoh lah. So if you are working in the lab, lepas ni kalau you jadi independent researcher, if you are working in the lab in whatever industries, make sure you know what kind of chemical that you are working for. Okay, it's very important. Dalam lab, it's not a playground, so you need to know semua chemicals, apa dia punya karakteristik, so what are the component dalam, so that you are aware, you are ready, so you know what are the precautions that need to be taken. Okay. Okay, so if there is damage DNA, kita ada repair system. Okay, in fact, kalau kita waktu kita run DNA replication, kita nak amplify DNA. Contoh buat PCR, we want to amplify DNA kan. We want to replicate the DNA. Zaman sekarang, it is a very so banyak, there is a lot of discoveries on bacteria that can produce DNA polymerase that have the proofreading characteristic. Maksudnya, that DNA polymerase can help to repair can prevent mutation ke something. Okay, ada one of the very popular zaman dulu DNA polymerase known as a Taq polymerase, TAQ. You boleh baca, Taq polymerase is isolated from thermophilic bacteria. Okay, daripada hot spring or something. Okay, this DNA polymerase. Yeah, so it is known as this Taq polymerase. Dia ada proofreading mechanism, so it can help to prevent any mutation ataupun boleh helps in mismatch ke untuk correct whatever problem dengan DNA. Okay, because mutations ni dia led to undesirable changes to gene. Okay, dia boleh mismatch, dia boleh add new nucleotide, even ada addition satu, deletion satu nucleotide, walaupun satu nucleotide dia akan terus bertukar sebab replication. Kalau you still remember the central dogma from replication into last kali kita akan ada translation. So even ada satu mismatch respect DNA, dia akan translate to different things. So kita punya badan tak boleh nak perform whatever roles that sequence carry. Okay, so it is very important. So that's why in our system kita ada all this repair system, but it may, even though it has been mended, even though it has been restored, even though it has been repaired, dia mungkin imperfect. Dia akan still produce mutation. Okay. Okay, so what is mutagenesis? Okay, I have explained this. Okay, we have so many environmental factors that affect, okay, that can lead to DNA damage. UV, that's why kalau nanti masuk lab, you nak kena UV apa, kita punya apa nama dia. So make sure you be careful lah dengan segala UV, ionized radiation, certain chemical agents kan, macam ethylene bromide, acridine orange, it may induce, okay, may lead to damage DNA. Okay, contoh. So how these factors, environmental factors, can damage DNA. Okay, for UV radiation, it promotes the formation of cyclobutyl ring between adjacent thymine residues on the same DNA strands. So it leads to the formation of abnormal kink in DNA helix. Okay, contoh DNA helix kita tahu kan, that double helix punya structure macam mana. So kalau ada UV radiation, dia akan form abnormal kink, dia akan jadi lain lah. Okay, so consequence dia, it will block replication and also transcription process. From replication, dia akan berlaku transcription and translation. So kalau replication and transcription pun dia dah block, apa lagi? Translation. So you cannot translate. Alright. Okay, for ionizing radiation, so how it damage DNA? It has a direct action on the DNA molecule. So it can damage directly on the DNA molecule or indirectly by inducing the formation of free radicals that led to strain breakage. Okay, kita tengok setiap factor so it has different mechanism how it damage DNA. Alright, for chemical mutations, okay, So what is chemical mutagens? Is a molecule, substances that damage DNA by inducing mutations, okay, which falls into two major classes. One is point mutation, and another one is insertion or deletions. So what does it mean by point mutation? Okay, so point mutation when there is one base pair replaces another. Okay, contoh kat sini lah. Okay, either by transition or transversion. Contoh, okay, the best example is on what happens the mutagenesis by nitrous acid. Okay, so what does nitrous acid do? Okay, on this mutagenesis, so it deaminates the cytosine, uracil base cytosine. Then deaminate cytosines so it produces uracil. Okay, and it base pair with adenine instead of guanine. Okay, so dekat mana? So it deaminates the cytosine, dia deaminates kat sini. Okay, and dia base pairkan dengan adenine. Okay, cytosine kita tahu dia kalau complementary dia sepatutnya dengan guanine. Okay, but when mutagenesis occurs, so this is what happens. So can you imagine macam mana dia nak, so what happened to DNA sequence kalau dah berlaku tak ada complementary base pairing. So how can it translate, kan? How can it translate how that specific sequence can be translated into protein? Okay, bila cytosine dah jadi uracil dengan adenine, patutnya cytosine akan complementary base pairing dengan guanine. Okay. Okay. Same goes jugalah kalau dengan adenine ni apa jadi? Okay, adenine patut dengan thymine. Dia dah tukar menjadi hypoxanthine and cytosine. So dia deaminates amino. Okay, dia deaminates cytosine and also adenine. Alright. For insertion deletion, okay, insertion deletion either addition of insertion of nucleotide or deletion of nucleotide, okay, daripada DNA. Contoh, okay, dalam contoh acridine dyes. Okay, so acridine dyes can intercalate DNA sequences. May arise from the treatment of DNA with intercalating agents such as acridine orange or proflavin or acridine bromide. As I mentioned, kan, bromide. Okay, so it can intercalate, okay, DNA sequence. So it's very harmful. Okay. Okay, contoh intercalating agents, acridine bromide, acridine orange. So it insert between, it can be inserted between step, okay, of that base pair in DNA. Okay, nampak tak contoh kat sini? So this is contoh, this is your double helix. Okay, when intercalating agents, okay, inserted, okay, in between express, okay, DNA ni, so it causes structural distortions. Okay, so that's why kadang-kadang kalau berlakunya mutagen DNA, so we are going to exhibit a very special, unique, different situation. Okay, kita akan exhibit macam-macam lah. Okay, that's why ada contoh orang OKU, there must be some problem with DNA sequence, okay, that led to maybe you can do reading, okay, on contoh lah kita ambil Down syndrome. So what happened to their sequence, okay, which part dalam DNA sequence dia yang different from normal people. Okay, dekat mana? Is it chromosome? Chromosome dia tak cukup berapa? Kalau normal kita berapa? So that it helps you to understand better on the what happened if we when we have changes in specific DNA sequence. Okay. Okay, so So that's the example of DNA damage. So we know that all this DNA error can be repaired, can be mended, okay, because we need to ourselves need to maintain the integrity of genetic information. Okay. Okay. So how DNA damage can be repaired? Okay, one of the method is by direct reversal of the image. Okay. Contoh, if we have this enzyme DNA photolysase, it helps to restore pyrimidine dimer to their monomeric forms. Okay, so either by this flipping, so this is how enzymes are also DNA bases as part of repair pathways. Okay, contoh is the reversible of base methylation by alkyl transferases. Okay. Any direct reversal of the image, okay, dia guna enzymes je. Okay, reversal of base methylation. Okay, another one is base oxidation repair. Okay, this one is for bases that can be repaired by removal or replacement. Okay, so this enzyme cleaves the glycosidic bond of altered nucleotide, leaving a deoxyribose residue with no attached base. So this one it produce AP sites. Okay. So then a glycosidase helps to alter the to produce the AP sites. That's how it repairs. Okay. Another one is position repair. Okay, so this one's specifically to repair the modified nucleotide lineages. Okay, example due to UV irradiation and chemical adducts. Okay, so in humans, this is a major defense against sunlight and tobacco smoke. Okay, by nucleotide repair. Okay, so how does it perform? So this enzyme endonuclease cleaves the damaged DNA strand at the 7th and at the 3rd and 4th phosphodiester bonds from deletions 5' and 3' sites. Okay, so it helps to nama pun oxidation repair kan, so it repair the nucleotide. Okay, so it cleaves the damaged DNA at the 7th and the 3rd and 4th phosphodiester bonds from deletions 5' and 3' sites. Okay, another one is mismatch repair. So it helps to correct mismatched nucleotides and is primarily for base mismatches and insertion deletion mismatch generated. Okay. So semuanya sangatlah penting untuk maintain fidelity of replication. Okay. So one thing that you need to know is many mutagens are carcinogen. So it can induce, it is carcinogenic lah. Okay, it can induce carcinogenic. Okay. So kalau you tengok ni kan, dia macam it remove one segment. Okay, it remove one segment of the damage, then a segment. Okay, dia remove and juga dia replace. So that's how the nucleotide acquisition repair. Alright. Okay. So now we move to the problem with DNA replication. Okay, ada soalan tak? Last topic. Tak ada. Okay, boleh dengar?
Boleh, Doktor.
Okay. Okay, so problems with replications, okay? Sekejap. Okay. Okay. So the ends of linear chromosomes, okay, it has problem, okay, with the ends of linear chromosome, okay, especially for the replication machinery. Okay. So what is the problem? Okay. Okay. Problem is DNA polymerase cannot synthesize. Okay. Cannot synthesize the extreme 5' end of the lagging strand. Take note. Okay, what is the problem with the replication? Okay, yang problemnya dekat end of linear chromosome. Okay, the problem is at ends of linear chromosome. So the problem is DNA polymerase cannot synthesize the extreme 5' ends. Sebab tu dia kata ends of linear chromosome. The extreme 5' ends of the lagging strand. Okay, DNA polymerase cannot synthesize. Even kalau RNA primer packed with 3' end of the DNA template, still dia tak boleh replace dengan DNA. Okay. So what happened is, okay, in the absence of mechanism, bila dah tak boleh of completing the lagging strand, normally linear chromosome dia akan shorten. Dia akan dipotong. Dia akan shorten at both ends by at least the length of an RNA primer with each round of replication. Okay, we know that the polymerase cannot synthesize extreme 5' end of lagging strand. So what happens is dia akan shorten. Shorten the linear chromosome. Okay, at both ends. Tapi dia ada disadvantage pula. Okay, so that mechanism kalau di shorten dia akan ada disadvantages. Okay. Okay. So how to overcome the problem? Okay. Certain cells exhibit telomerase activity. So this is an enzyme. Dia ada telomerase enzyme. Okay, so what is telomerase enzyme? So these are RNA dependent enzymes that extend the telomeres of chromosome. Okay, kan kita tengok what is telomere. Alright, so what does this enzyme do? It induces the formation of the telomere DNA at the overhanging 3' end. So it helps to adding nucleic acids to the 3' hydroxyl group. Okay, so when enough telomeres have been added, primer is added, and DNA polymerase will transcribe the telomere strand similar to the action already described for consecutive fragments. Okay, bila DNA polymerase cannot synthesize at the extreme 5' end of the lagging strand, so we have this enzyme telomerase. So telomerase helps to synthesize telomere. Okay, dia helps to synthesize telomere. Okay, dekat lagging strand. Okay, bila ada telomeres, primer is added, so DNA polymerase boleh transcribe this telomere. Okay, sama. Okay, macam di describe for Okazaki fragments. So what is telomere? Okay. Okay, the end of eukaryotic. Okay, this one is specifically for eukaryotic system. Okay, the ends of eukaryotic chromosome. Okay, okay, ataupun kita panggil dia telomeres. Dia ada unusual structure. Apa maksudnya unusual structure? Okay, so the DNA sequence of telomere. It consists of hundreds or thousands of tandem repeats of a short G-rich sequence. Okay, dia unique. Okay, dia ada unusual structure and unique. Yes.
Ulang part DNA sequence kat dalam ni ada tendon apa doktor?
Okay, tandem repeat. Maksudnya dia ada repetition. Okay, it consists of hundreds or thousands of tandem repeats. Maknanya dia ada repetition of a short G-rich sequence. Short G-rich sequence. Okay, at the 3' end of each chromosome. Okay, so end of linear chromosome dia ada ni. Dia ada di telomere. So telomere dia punya sequence macam mana? Dia ada, contohnya kalau telomere ni dia ada hundreds, contoh hundreds, katalah 500. So 500 ni dia adalah terdiri daripada repetition. Katalah dia ada TTAGGG, dia ada short G-rich sequence kan. So dia ada repetition sampailah di hundreds. Thousands of repetitive regions, okay, of this short G-rich sequence. Okay, so this one dalam human lah, TTAGGG. So this is the telomere sequence dalam human. Kalau dalam prokaryotik, kalau dalam protozoa dia different sikit, dia ada TTGGGG. Okay, dia different lah. So this one specifically for human. Okay, so ini yang membezakan. Kalau when we talk about telomere, you should know this unique characteristic. Dia punya sequence macam mana. Okay, so what? So why is it special to have this GC rich? Okay. And this telomere DNA is synthesized and maintained by an enzyme telomerase tadi lah. Okay. So kita tahu kan telomerase is a RNA dependent enzyme. So basically it's a ribonucleoprotein. It's a complex of RNA and protein. Okay, enzyme telomerase. Okay. So it acts as a cap that protect against degradation of the chromosome by overhang. Okay. Okay, kenapa dia ada this repetitive short G-rich sequence? Because dia ada dia punya special characteristics. Okay, because it's located at the end of the linear chromosome. Okay, so it needs to have this repetition of this short G-rich sequence to help to protect against degradation of the chromosome. Dia akan berlakunya overhang. Dia akan ada overhang kat sini. Okay, dia akan ada overhang kat sini. Because DNA polymerase tak boleh nak synthesize dekat end of extreme 5' end. So dia akan ada overhang 3' end kat sini. Okay, dia akan ada overhang 3' end kat sini. So dekat sinilah telomerase. Dia akan help the formation of telomere DNA dekat overhanging. Okay, telomere ni akan disynthesize oleh telomerase dekat overhang, this 3' overhang. Faham tak? Bila DNA polymerase dia tak boleh synthesize dekat extreme 5' end, dia akan ada overhang dekat 3'. So telomerase helps to synthesize telomere dekat 3' overhang. So bila dah ada telomere, so DNA polymerase akan boleh synthesize macam biasa. Okay, so DNA polymerase once telomeres have been synthesized dekat 3' overhang, primer added, so DNA polymerase can transcribe the telomere sama macam Okazaki fragments. So that DNA replication is complete. So at the end of the linear chromosome, you need to understand what is the problem. So how it overcome by having this telomere, this telomerase. Okay, faham tak? Ada soalan? Okay. So however, sooner or later the telomeres will be used up and the chromosome will start shedding important genes. Okay, when this happens, the cells will usually die. Okay, towards the end, bila kita umur pun dah meningkat, okay, all these telomeres, dia punya mechanism, system is not that efficient as umur kita sekarang. So it start to use important genes. So that's why kita akan Tak adalah lepas tu. And many cancer cells have shortened telomeres and telomerase is active on these cells. Okay, take note on that. Okay, so you can read this. Okay, so for this specific topic, you should understand what is the problem at the end of the what is the problem with replication, what is the problem with the at the end of the linear chromosome. So how the problem is overcome. So apakah yang diperlukan untuk overcome the problem? Okay, by having telomerase and telomeres. So understand what is telomere, understand what is the role of telomerase. So this topic should be straightforward lah, senang je. Okay, telomerase and aging. Telomere length is related to aging. Okay, all the subjects by and large have shorter telomeres compared to youngsters lah. Okay, so short telomeres appear to be particularly associated with cardiac age. Okay. Okay, other factors also affect telomere length and is subject of much ongoing research. So it's another interested field, okay, research field that can be explored on the telomere, on the telomerase. Kalau you cari, if you try to search to search journal on telomerase, telomere, you can see there are so many research have been conducted, okay, for this specific molecule, okay, telomerase, telomere, and how it related to aging. And you also can try to look on the associated of telomere with cancer. Okay, nampak tak the importance of telomere? Okay, telomere dengan aging, dengan cancer, and also with DNA replication. Because we know that replication is very important for maintaining kita punya genetic information. Alright. Okay, so you can have a look at this. Saya dah compile kan satu what happened during at the end of the linear chromosome bila DNA polymerase cannot synthesize at the 5' the extreme 5' end dekat lagging strand ni. So telomerase helps to synthesize telomere, so it maintains telomere so that DNA polymerase can synthesize new DNA strand. Okay, ada soalan? We are done with DNA metabolism. Before we continue with RNA metabolism next Wednesday, ada soalan?
For now, it's okay.
Okay. Lain-lain? You all jangan keluar lagi. Saya nak snap for attendance.
Tak ada doktor.
Alright. So we have another two topics. Next Wednesday kita start dengan RNA metabolism, then last one on the gene expression. Siap saya lah. Kita tengok balik. Okay. Ada soalan? No?
Doktor,
Ya.
tapi doktor nak tanya pasal lab punya lab.
Okay.
Pasal calculation doktor.
Okay, kenapa calculation?
Okay. Macam group saya dia buat dilution kan. So tambah 10 ml air kan. Dilution factor tu kali dengan absorbance tu dulu ke atau formula lepas tu kali dengan 10? Sebab macam tak logik doktor dia punya results tu.
Tak logik kenapa?
Sebab macam saya kalau andai kata blood serum divided by standard times 30 times 10 kan. Tapi group saya dilute kan dengan 10 ml air. So kena times 10 lagi kan. It become 5,000 something. So macam tak logik lah untuk uric acid concentration to be that high, right?
Okay, kalau you tak dilute, kalau you calculate pun sama kan, betul tak? Kalau you tak dilute,
Kalau tak dilute, dia jadi, alamak tak ingat lah berapa result dia, tapi macam bacaan dia lari lah.
okay, mungkin you boleh direct dengan saya nanti. Okay, saya explain on the
Direct dengan doktor?
calculation. Okay?
By WhatsApp ke atau pergi jumpa doktor kat office?
By WhatsApp pun okay.
Oh, okay.
Alright. Okay.
Thank you, doktor.
Alright. So ada soalan lagi? Okay, if there is no, doktor nak hard copy ke? Yes, I need a hard copy. Lab report. So lab report, okay, saya biasanya saya nak individual, but this time I agree for you to submit dalam group. Okay, but bila dalam group, I expecting more. Okay, in terms of marking point, will be more strict lah sebab you bekerja dalam group. Okay, kalau duduk individual, I'm more lenient. Okay, because I would like to train you on how to do a report, kan? Report macam nak buat discussion, maybe result sama, but I would like to see how do you discuss, how do you present your results, okay, how do you elaborate, okay, but this time bila, okay lah, but I agree untuk you hantar dalam group sebab kita ada 2 je, but I'm expecting more, okay, on the discussion, on presenting results, okay, because we should take it lab report seriously because this is how, because you are going to apply this into you punya FYP thesis, okay, so I'm going to be more strict lah dalam marking untuk amali, okay, hard copy lah, hard copy, kalau tak, kalau okay dengan you all, saya do we have lab this Wednesday? Yes, ada lab, right? Okay, ada soalan?
Doktor,
Okay, if there is no, yep.
Rabu ni online tak pagi?
Kalau dah selesai ke yang positif semua tu? Bila ada yang positif tu yang saya tukar pergi online.
Oh, okay.
Dr. Dina inform saya kan, because we need to know the status test tu. So biasanya we all akan communicate each other bila ada case macam tu. So okay, kena buat online lah. Okay, hopefully by next week lah kot. Kalau berkesempatan kita buat physical lah, kalau semua okay. Okay, no other questions. So I'll see you next Wednesday. So online jugalah for the time being. Okay. Okay, thank you very much everyone. Terima kasih.
Thank you,
Ada
doktor.
soalan?
Thank you, doktor.
Thank you.